Please join me in welcoming our speakers. Thank you, Cheryl. It is wonderful to be here and discuss with you some of the research in the stem cell field that has been going on, and tell you about where things stand right now and what directions we are headed towards in the future. So as many of you know, this field has been swamped with a lot of controversies and dilemmas and debates, not just from a scientific, technological standpoint, but also from an ethical standpoint. So in order to understand where all those issues are stemming from, I should first try and understand the science, the biology of stem cells, and what sort of potential they hold, and what they've been used for so far, and what they can be used for. So let's look at that. So when most people these days talk about stem cells, generally referring to embryonic stem cells, so what are embryonic stem cells and where exactly do they come from? To answer those questions, let's take a look at early embryonic development, or the developing embryo. Right from stage 1, we start with the fertilized egg. This tiny cell has the potential to develop into a full-grown individual with many different cells, many different types. And it starts off by dividing into 2 cells, which go on to becoming 4, 8, and after a few cell divisions, we get a small, compact ball of cells, which we call the morula. Now the fun starts. The outer cells of the morula differentiate into a different cell type, which forms the placenta, which is a tissue that that helps attach the embryo to its mother. And this structure is called a blastocyst, and it's basically kind of like a hollow ball of cells with a pretty large cavity and a few remaining cells inside. And it is this structure from which embryonic stem cells are derived. So let's take a closer look at the blastocyst. So as I said, the outer layer makes an extra embryonic layer, which in our case is the placenta. And not surprisingly, the cells in this cavity are called the inner cell mass. And these cells are responsible for giving rise to the entire individual, or the whole embryo. And as developmental biologists like to say, the embryo proper. Now, if we take these cells from the inner cell mass and put them onto a dish and give them some nice nutrients and growth media, these cells can grow in culture in the lab, and we get embryonic stem cells. Very nice. We have dishes of cells growing in the lab. What do we do with them? What are they good for? One of the properties, so-called hallmark properties of these embryonic stem cells is that of self-renewal. So this means that we can grow them pretty much indefinitely in the lab, in these dishes, and they seem to retain their same shape or form and their properties. And the other stem cell hallmark, or really cool property of embryonic stem cells, is that of pluripotency, which is the ability to give rise to all different kinds of cell types that are present in an individual. So under specific conditions, we can induce these embryonic stem cells, or ES cells as we like to say, into pancreatic islet cells, which secrete insulin, cardiac muscle cells, or neurons. So I hope that you can begin to appreciate that the similarities between the cells of the inner cell mass and embryonic stem cells the fact that the cells of the inner cell mass can give rise to an entire embryo, and ES cells can give rise to any tissue type or any cell type present in an embryo. And these similarities can be noted at the genetic level as well. So we know of certain genes like OCT4 and NANOG, which are expressed in the inner cell mass in the embryo. And these are extremely important or key genes in regulating embryonic development. These same genes are also expressed in ES cells and are vital in maintaining embryonic stem cell pluripotency. So how do these genes work? Well, these are what we call transcription factors. So what transcription factors do is they bind to various places in the genome or in the to the DNA, and by doing so, they can activate gene expression, or under different conditions, they can repress gene expression or transcription, as we say. So much of the current work is focused on how OCT4 and NANOG do so, and which genes in stem cells and in the inner cell mass they activate or repress. So if we have a good understanding of that, you can imagine play around with that system and induce or interfere with that regulation and direct embryonic stem cells into becoming pancreatic islet cells or cardiac muscle cells or what have you. That's all nice to say, but what have stem cells been useful for so far? Well, human embryonic stem cells have been, they were isolated not too long ago, it was in 1998, and still working on how to get various kinds of tissues from that and see what we can do with those cells. But mouse ES cells were derived in 1981, so practically 25 years ago. And they've been useful for geneticists for a very long time, engineering transgenic mice, which have been key in understanding various modeling human diseases and doing mouse genetics. So how do you make a mouse transgenic model? Well, we start with two different kinds of mice. Say for our purposes, we have a pink mouse and a blue mouse. We get pink ES cells from the pink mouse, and our blue mouse, who's the egg donor, donates a blue embryo, which goes on to becoming a blue donor blastocyst. Now, we have a piece of foreign DNA, and what we want to do is make a transgenic mouse that carries this piece of foreign DNA, which encodes your favorite gene, in every cell of its body. So what we do is we take this foreign DNA and in a little tube, mix it with these pink ES cells, shake it up, zap it a couple of times with electricity. And this zapping procedure is known as electroporation. And what it does is it allows the foreign DNA to enter these cells. So once these cells are electroporated, we inject them into this donor blastocyst. So this new blastocyst has embryonic stem cells of different kinds, pink and blue. We let the blastocyst develop, implant it into a surrogate mother, and what we get out of it is a chimeric mouse. So this chimeric mouse has been derived from this mixed stem cell blastocyst. So the pink stem cells contribute to some of the tissues, and the blue blue ones contribute to others. We get a pink and blue mouse. And we hope that some of the cells, or some of the pink stem cells, which are carrying a foreign DNA, gave rise to the germ cells being the egg and the sperm. Why? Because if we now cross this chimeric mouse to a blue mouse, we can get, but the chimeric mouse still has those germ cells derived from the pink cells. We still get a pink transgenic mouse which carries the foreign DNA in every cell of its body. And you can see how useful this technique is in studying how a particular gene could affect development, or introducing a different gene into the mouse to see what effects it has. So we can make transgenic mice. It's wonderful. What do we do with human embryonic stem cells? Wouldn't entirely be ethical or very nice to make human transgenics. But we can still use transgenic technology or stem cell technology in studying human ES cells, and what they can do. So if we have a piece of foreign DNA, we can electroporate it into human ES cells, just like we did with mice, and see how that affects differentiation of these embryonic stem cells. So using this technology, genetic engineering, we can examine which genes maintain the identity of these stem cells, and how we can manipulate these stem cells by introducing foreign DNA into and direct differentiation of these into neurons and cardiac muscle, or whatever you'd like. So that's great. We can get neurons or skin tissue on a plate. How is that useful? Well, you can imagine a scene where you have a patient who's suffered from massive burns, say. And with the current technology we have, this patient would receive skin engraftment from skin donated by some kind donor. But there's a problem. Our immune system recognizes self from non-self, which means that our immune system has the ability to tell whether this tell apart the cells of our body from other cells from somebody else's body. So it's only a matter of time, and this graft is rejected, and we're back to square one. So the idea of stem cell therapy is that you can have patient-specific stem cells. So this guy can have his own stem cells, and so to overcome the problem of graft rejection or immune rejection, as one may call it. Now that I've said that stem cells are derived from the blastocyst, how in the world do we get a blastocyst from a full-grown human being? Here's how we propose stem cell therapy would work. You could get it would work through a procedure called nuclear transfer, or more popularly known as cloning. So we would get a somatic cell, for example, a cheek cell, from the inside of the cheek of the patient, and so this somatic cell from the patient would carry, or the nucleus of this cell would carry his particular genome, and we get an egg from another donor, and take the nucleus out of that egg, and we take the nucleus from the somatic cell from our patient and transfer it into the egg obtained from the donor. What does that do? So a somatic cell and a fertilized egg are very different cells, as you know. They're different because they express different genes. So genes that may be turned on or turned off in a cheek cell or somatic cell may not be turned on or turned off in a fertilized egg. they differ. The somatic cell and fertilized egg differ at what we call the epigenetic level. What we mean by this is they have different methylation patterns. So our DNA is modified at various places with this mark called a methyl group. And the presence or absence of these methyl marks can dictate whether a gene is turned on or not. And so the methylation patterns of various genes are extremely different in a somatic cell versus a fertilized egg. So due to these differential methylation patterns, you get differential gene expression. And by nuclear transfer, the environment of an egg would reprogram the genome of a somatic cell into that of a fertilized egg. So it would behave like a regular embryo. Now we have a regular embryo, we hope, from our patient. And we let that develop to the blastocyst and get patient-specific ES cells. We hope that we can differentiate those into, in this case, skin tissue and implant it, and that would be pretty dandy. I hope that in 20 years from now, this is where my story would end. But unfortunately, today we're faced with a lot of scientific and technical issues as well as ethical dilemmas surrounding this work and stem cell research in general. And I'll hand it over to Chris to discuss more on that end and tell you what's going on and how we're tackling that.
Thanks, Divya. So I'm going to discuss a little bit about the problems with cloning as it stands now, at least in humans, and therapeutic cloning in particular. But before I start, does anybody have any questions about this concept here? So feel free to interrupt at any time with any questions. But this concept here that the nuclear transfer procedure removes these methyl marks and thereby makes the DNA in the embryo and in the embryonic stem cells more likely resemble that of the normal fertilized egg. This basically is the critical concept in all of cloning and in all of the stem cell field. So basically, the process of nuclear transfer just acts so that the adult cell's nucleus, the adult cell's DNA, can be exposed to these demethylating activities that we don't yet understand. But it's the activity of these demethylating enzymes or whatever factors they may be that basically turn the adult cell into the embryonic stem cell. Yeah?
I will ask one question.
Sure.
You found other ways to separate the methyls off. Would that eliminate the political nature of this problem?
I think she wants you to speak. Oh, could you repeat the question? I'm sorry.
If you found some way to sever these methyls other than embedding them in a fertilized egg,
Right, okay.
would that alleviate the political aspect?
Yeah, it would totally. So the question is, if we could bypass the use of the egg and remove those methyl groups in some type of artificial manner. So there are chemicals that do that, but the problem there is that the chemicals have a lot of side effects, and that doesn't really seem to be a viable option as such. There's also a lot of labs whose goal it is to understand what those enzymes are. So if we can someday, hopefully soon, figure out what those enzymes are and then use them at will to perform that procedure, then we could circumvent the use of the egg in the nuclear transfer procedure. But I'm going to talk more about that in a second. Yeah.
As a follow-up, does this mean
Hold
that
on just a second. Just so Wester can hear you.
Hi, does this mean that the methylating patterns are what allows the body to know self from non-self as well?
No, that's sort of another issue that involves the immune system and a whole stack of textbooks like this big, so we won't really get into that. But the methylation patterns basically establish gene expression patterns, and every different cell type in your body, be it a skin cell or a heart cell or what have you, has a distinct methylation pattern. the embryonic stem cell has sort of a blank methylation pattern that will subsequently allow it to become any of those different cell types. So the immune response is sort of tied into that a little bit, but it's a kind of a more complicated issue. So with that, I guess I'll get to where Divya left off here. So what I'm going to talk about is obstacles to the generation of patient-specific ES cell lines for therapeutic use. So I think everyone is in agreement that no one wants to clone humans to make copies of ourselves. It's basically strictly, the reason we do all this research is strictly to generate patient-specific ES cells that have already been proven in the lab that human ES cells can differentiate into pretty much any cell type in your body: neurons, skin, bone, muscle, et cetera, et cetera. So the process of differentiating the ES cells into the desired tissue is really not a rate-limiting step at this point. But the rate-limiting steps are sort of as follows. The lack of a plentiful supply of oocytes, or human eggs. Of course, the moral and ethical issues that surround the creation and destruction of human blastocysts, and the inefficiency of the nuclear transfer procedure, including technical and biological limitations. So I write inefficiency of nuclear transfer here because in mice it's very inefficient, but as we know now from this scandal with the Korean papers that came out, in humans there's zero efficiency. No one's been able to do it yet so far. So it's theoretically possible that it will never work in humans. There are some species that are for some reason particularly resistant to cloning, but everything I'm going to talk about we've done in mice, and in large part the mice mimic the situation in humans for a lot of things. So the question is, can we overcome these obstacles to make ES cell-based therapy a reality? Oops. Okay. So the first and one of the easier issues to address is the supply of human oocytes. So one thing that people do now, and what the Koreans have done and what people that are into therapeutic cloning do, is increase the yield of oocyte isolation by giving the female drugs to superovulate. So these are usually nasty hormones, and they have all sorts of side effects. So this is really not a desirable thing to be doing. A lot of people that undergo treatment with these hormones have real nasty side effects, including ovarian hyperstimulation, causing hyperproliferation that can, in some rare cases, lead to ovarian cancer. So solutions to this dilemma here include achieving sufficient efficiency of cloning with a small number of eggs, such that you would avoid having to use the superovulation procedure. And something I'm going to talk about in more detail that ties into some of the other dilemmas is circumventing the use of the oocyte at all, or altogether. Okay, the second bottleneck here is the moral issue. So can ES cells be derived in a morally acceptable manner that's morally acceptable to everyone? Two recent studies that just came out last year have kind of, actually early in the beginning of this year, have begun to address that. So the first one that I'll talk about came from this company called Advanced Cell Technology in Worcester, Massachusetts, where I guess we're broadcasting to. And what they have done, so Divya talked about the stages of early development from the fertilized egg or the zygote to the morula to the blastocyst, and of course the blastocyst can then implant in the uterus to give rise to an embryo, or the embryonic stem cells can be harvested from the blastocyst, and these cells can be differentiated into whatever cell type you desire. What the people in Advanced Cell Technologies have done is they have been able to take a single cell, basically physically plucking this cell from the morula and then attaching this cell to pre-existing ES cells from another source, thereby nurturing the growth of this individual cell without destroying the embryo. So removing one cell, generating ES cells from that, and allowing the rest of the morula to develop into the blastocyst. So if this were done in an in vitro fertilization setting, for example, this blastocyst could then be saved and implanted, so then you could theoretically, have a child and have ES cells that are specific to that child. So this technology has now, the principle has been proven that it is possible, but again, it remains very inefficient, and a lot of times the embryo, I believe, I'm not sure on this one, but I think a lot of times the embryo itself is destroyed. So there's efficiency issues with this procedure. Okay. So just some pictures on how that last procedure actually works. These green cells here, these are embryonic stem cells expressing a green fluorescent protein that had previously been isolated, and this stem cell that was plucked from the morula, you can see is growing as a non-green cell on the side of this colony. So what these folks have done is grown this new cell derived from the morula alongside with other ES cells because they support one another's growth. They don't like to grow by themselves. So by doing this, it supports the growth of that single cell. And then they've been able to isolate these cells, and this is the blastocyst injection procedure. So that's a blastocyst right there, and here you can see these morula-derived stem cells going into the blastocyst, and these blastocysts are then implanted into a foster mother, and they then give rise to children. So what you can see here is some of these embryos are blue, and that's because the non-green cells, i.e. the morula-derived cells, contained a gene that marks them blue. So we have green and blue, and what you can see here is that these blue embryos are then derived from these morula cells. So this test acts as a proof that these cells are, in fact, pluripotent stem cells because they are capable of giving rise to an entire organism. So again, this is all done in mice. Any questions? Pretty clear? Okay. Okay, the second possible way to circumvent some of these ethical ethical concerns about human cloning is this procedure called altered nuclear transfer. And this was originally proposed by this fellow named William Hurlbut in Stanford University. He's a biomedical ethicist at Stanford. And the idea goes like this. Altered nuclear transfer proposes to produce pluripotent stem cells without the creation and destruction of human embryos and provide a technological means to bypass current ethical, legislative impasses, et cetera, et cetera. So how does this work? So we call this ANT. Altered nuclear transfer uses nuclear transfer, but with a preemptive alteration to the blastocyst to ensure that no embryo is created. And this is done by altering or silencing or deleting genes essential for, and as he states it here, the integrated and coordinated organization that characterizes a living being. Therefore, the unorganized trajectory of organic growth would still create an inner cell mass where you could derive embryonic stem cells, but couldn't create the tissues that support the embryo, i.e. the placenta. So Alex Meissner in our lab last year went about to prove that this principle would actually work. And let's see if I can walk you through this. Okay. So under normal cloning procedures, therapeutic cloning versus reproductive cloning, you have adult cells. Here we term them tail-tip fibroblasts, so these are adult skin cells. The nuclei of these cells get injected into an oocyte, and this is, of course, the cloning procedure. You then have a cloned blastocyst, termed here nuclear transfer blastocyst. This blastocyst could then be implanted for reproductive cloning, which we're all in agreement is not good. But the cells of this inner cell mass could be harvested to generate new embryonic stem cells, but thereby destroying this blastocyst, which could potentially become a human life, which is therefore morally questionable. So what Alex did was he took these tail-tip fibroblasts, and then using a virus, he infected these cells with a virus that was able to basically stop the expression of a gene that's required for the formation of the placenta. So as Divya mentioned earlier, the outer cells of the blastocyst form the placenta and the extra embryonic tissues, while the inner cells of the inner cell mass are or can become the embryonic stem cells and can be grown in culture. So by knocking down or inactivating the gene that can form the placenta, basically you are left with something called an ANT, or an altered nuclear transfer blastocyst. So again, he knocks down this gene in the adult skin cells, clones those cells, and has this altered nuclear transfer blastocyst. So the idea here is now that you have this blastocyst, or an ANT blastocyst, that still has cells of the inner cell mass that can become embryonic stem cells, but because it lacks the genes that are necessary for formation of the placenta, it cannot implant into the uterus of the mother, and therefore cannot form a life, in this case a mouse life or theoretically a human life. Therefore, the argument goes that this blastocyst is in fact not a blastocyst and does not represent a human life because it does not have the properties necessary for the formation of an organism. So this actually works remarkably enough, and here's some images, and what you can see here are these, I believe, are the blastocysts generated from the knockdown. This gene is called CDX2, the gene required for formation of the placenta, and these are the controls. And what you can see here is that once you've knocked down this gene, you can see the virus has a green fluorescent marker in it, so you can see that the infected cells are green, whereas the control cells are not green. And when you allow these to grow to form blastocysts, the wild-type blastocysts will implant in the uterus and give rise to children, but the blastocysts with the knockdown of the CDX2, the placenta gene, cannot do that. So this works in mice. But yet there's still controversy whether this is ethically okay or not, of course. But we won't get into that. So what we've talked about so far are these various ways to get to human ES cells. There's the classical ES cell derivation method. There's the ES cell derivation method of the single blastomere, which is the advanced cell technology method. There's nuclear transfer, and then the altered nuclear transfer method, which I just spoke about. So the question is, can we simplify this whole scheme and get around a lot of these things? So can we reprogram an adult cell and an adult genome back into an embryonic state without having to use the oocyte and without having to expose it to the factors, the unknown factors within the oocyte? So that's the real question. So again, the critical point here is that the oocyte contains these as of yet unidentified reprogramming factors that can strip the adult genome of its methylation marks and thereby allowing it to re-express the genes that Divya spoke about, OCT4, NANOG, and a few others. And it's the activity of these genes that maintain the state of stemness in the stem cells. So the ES cells themselves contain these reprogramming factors. So can we harness these factors to reprogram the adult cells? So one approach to that is through the use of this cell fusion technology. And this was work done by Kevin Egan's group at Harvard, who also came from our lab at the Whitehead, but now works with human ES cells at Harvard. So this work was actually done in humans. And what he has shown now is that you can fuse an adult cell with a pre-existing, for example, a government-approved embryonic stem cell line. And when these cells fuse, the factors within the pre-existing ES cell line will reprogram the new adult cell back into an embryonic state. And that goes a little bit like this. So here's fibroblasts isolated from a person and previously isolated embryonic stem cells. Through the procedures that the transgenic procedures Divya spoke about, we can make these cells green, and we can make these cells resistant to this drug, pyromycin. And you can then fuse these two cells together by a chemical-based method, and what you're left with is a hybrid cell. And then you have to co-select for the two markers, so the gene and the drug resistance. And what you can see here is just a cellular analysis of the intensity of the green protein, and what you can see is that the fibroblasts have no green, the human ES cells have green, and the hybrids also have the green protein. And basically, this seems to work pretty well. The resulting hybrid looks a lot like a normal human ES cell colony. It expresses the pluripotency gene OCT4, which you can see here in red, and it expresses the GFP gene found in the donor ES cells, and it's also puromycin resistant, meaning that it's gotten the drug resistance from the adult fibroblast. So this is great. This works. It's not that efficient, but it's a solution, except there's one critical problem here, is that you still have you have 4 sets of genes. So you have the 2 sets of genes from the original ES cell, and then 2 more sets of genes from the adult nucleus. So this hybrid basically has 2 nuclei. So for any therapeutic purposes, the trick is really to destroy the nucleus from the embryonic stem cell while retaining the nucleus from the donor fibroblast from the adult patient. And right now, we have no clue as to how to do that. So that's clearly a limitation with this approach. So the last thing I'm going to talk about here is what I work on, and a lot of us in the lab across the river here work on, and that is genetic reprogramming. So can we recapitulate the effects of the reprogramming factors found in the oocytes and in the embryonic stem cells without using the oocytes or the embryonic stem cells? So let's simply reactivate these pluripotency genes directly in the adult cells through genetic manipulation. So as Divya spoke about, OCT4 and ANOG are 2 of those genes that we know are critical for maintaining the pluripotent state in the embryonic stem cells. So the idea here is that we can engineer adult cells to contain these 2 genetic constructs. And basically, to make a long story short, you can engineer this into a mouse, just like through the schema that Divya showed. So all the cells of the mouse contain these 2 genes, these trans genes, and that when you feed these mice this drug called doxycycline in their drinking water, very non-invasive, very simple, it will reactivate the expression of OCT4. So OCT4 is the pluripotency gene. It's only active in embryonic stem cells, not active anywhere in the adult. And if we turn this thing back on, can we now turn adult cells back into stem cells? And, okay, some graphics there. So Conrad in our lab showed last year that there is an interesting effect of OCT4 when you reactivate it in the adult. You're not, I'm sorry about the images here. You don't really get the adult cells turning back into stem cells, but you kind of get halfway there. And basically the phenotype of these mice, and particularly this is looking at the intestine of the mice, is when you feed the mice this drug called doxycycline, you can see here in brown staining for the OCT4 gene. No OCT4 in the normal mouse, but OCT4 comes on everywhere in the intestine, even after a short 2-day treatment with this drug. And what you can also see happening is there's an expansion of the cells that are dividing. This is a marker of dividing cells. And more interestingly, you get a complete shutdown of genes that are associated with a differentiated state. So these cells in the intestine normally are digesting food, secreting enzymes, secreting protective mucus, et cetera, fighting bacteria. They have a whole host of functions. But when you turn on OCT4, all that shuts off. And basically they sit there in a quasi-pluripotent state and divide. So just by reactivating one gene, we're already a small step towards genetically reprogramming adult cells. But we're not there yet. These cells are not embryonic stem cells by any means. They don't exhibit the properties of embryonic stem cells, but they are unable to differentiate into the normal differentiated cell types. So ultimately the question is, can we overcome these obstacles to make ES-cell-based therapy a reality? And maybe someday we can, but not without support for stem cell research. So I'll leave you with that. Thank you.
Now we'll have some time for formal questions. If anyone has a question, please raise your hand. Yes?
We've heard a lot about the encoding from the 4 base pairs of DNA to the 20 amino acids. Have you made any progress on understanding the encoding of the regulatory genes or isolating regulatory genes that control the differentiation of the methylation of the gene during this progression? And can you say something about that?
Yeah. So, well, there's two points there, I guess. The genes that control the differentiation of cells towards skin or bone or hair, there's a lot of them, and they're studied by hundreds of labs all around the world. And a lot of them are transcription factors, and there's usually down every pathway, whether it be the bone pathway or the brain pathway, there are what they call master transcription factors that can sort of drive that whole program of gene expression that activate all these downstream tissue-specific genes. But in regards to the methylation itself, that's a little bit more tricky of a question. So we do know what genes place the methyl marks on the DNA, and we know other genes that code for, well, we know the genes that code for the proteins that place the methyl marks on the DNA. And we also have identified and isolated genes that can then visualize those methylation marks on the DNA and bind to it and repress the transcription of those methylated genes. But again, the big black box there is, if it exists, the gene that codes for the protein that can then remove those methylation marks. So it's really the removal of those marks that allows us to go backwards in development, or potency, if you will. Going forwards and driving differentiation is fairly, not totally understood, but fairly well understood, and we are able to do that towards any number of cell types. Does that answer your question at all? Okay.
Do we have any questions from Worcester?
How are you, Cheryl? Let me thank both of the speakers for excellent presentations. Yes, we do have a question.
The embryonic fluid, couldn't they use a spectrometer to find out what the growth factors were throughout the period of development, so they'd have an idea of what was triggering different points of development in the genome?
So if I think I understood that as, Divya, did you understand that one?
I think they're asking what growth factors direct differentiation during development of the embryo.
Is that right?
Is that right?
So I don't know if your microphone that's on.
Yeah.
Well, assuming that is the question, do you want to answer that one?
Sure. Assuming that's the question, the answer is that we do know of certain genes that direct differentiation from the inner cell mass into whatever the next tissue type is in the differentiation hierarchy. And as I mentioned, OCT4 and NANOG are a couple of those genes. We don't. Sorry. Was that not the question, or? Anyway, so carrying on, we do know of those few genes, but there are still some others that need to be identified with respect to differentiating stem cells into all sorts of different tissue types.
It's become popular to save umbilical cord, bloods or stem cells or what
Sure.
have you. Can you say anything about, the potential for that?
Okay. Again, this is not my area of expertise, but my understanding is that the stem cells that are derived from cord blood are not pluripotent like embryonic stem cells. So they can what they are good for, though, is if, for example, you get leukemia and you need radiation, they are good for repopulating the immune system and the bone marrow and the types of things you need bone marrow transplants for. Those cells are, I believe, good for that. And I don't know if they've ever been actually proven to work in humans, but I think in mouse model systems that they are good for reconstituting the blood system and the immune system. But again, you're probably not going to get brain cells and neurons and more specific pancreatic islet cells out of the cord blood.
Well, that said, there is work that's going on which is trying to show that these cells they're not pluripotent, but they're so-called multipotent. So they can give rise to not all different types of tissues in the individual, but many. So you may not get neurons, but you may get 6 or 7 other different kinds of cell types.
Yeah, so I guess one way to look at it is if you look at a totipotent or pluripotent embryonic stem cell, and then you can imagine differentiation into various cell types as some sort of pyramid scheme, that cell types that are closer in one branch of the pyramid, for example, bone, muscle, cartilage, fat, all come from one common precursor, whereas a lot of other cell types come from. So there's hierarchical structuring of differentiation. So cord blood could maybe become other closely related cell types.
Do we have any other questions from Worcester?
Mary, go ahead.
Technology.
No. You know anything about that?
Could you hear that, Sean?
No.
Hold on.
I'm just wondering if there are any human cells that can be totipotent, like the neurospheres. I mean, can they be totipotent, not just pluripotent?
Well, there's a difference between totipotency and pluripotency, which we haven't really talked about at great length. Totipotency is the ability to give rise to an entire embryo, whereas pluripotency is the ability to give rise to all the different cell types present in an embryo. Those are two different things. The only totipotent cell, per se, that we know of is a fertilized egg. Because if you think about it, a fertilized egg can give rise to the entire embryo and also all the extra embryonic cells, like the placenta, which are key to development or formation of an individual. Whereas embryonic stem cells are pluripotent. They can give rise to all the tissue types of an embryo, but they don't give rise to the placenta. So you cannot form an entire individual from a single embryonic stem cell or even a group of embryonic stem cells. Neurospheres are, as we say, multipotent or...
Neurospheres, I guess, similar to the example in the blood, is that a neurosphere is a progenitor cell for neural cell types. So they're pretty limited to cell types in the brain. A neurosphere can't give you a bone cell, for example.
So we don't have any other cell type apart from a fertilized egg that can give rise to an entire individual on its own. Does that answer the question?
Yeah.
Any other questions in here? No? Worcester, do you have anything else?
No ethical debates, that's good. Yeah, so...
Could you just repeat that?
Oh, so the question is, most of the stuff I've showed here is being done in mice, and what's the sort of state of the art in the human. So after this, after the paper was published by the Koreans that it was possible to clone humans, everybody else working on human stem cells at that point sort of shifted their focus, I guess, a little bit, assuming that that barrier had been crossed. So since the scandal has sort of surfaced, people are sort of backpedaling, and the two human ESL research institutes that we're kind of in contact with are the Harvard Institute, where they are now, while they were doing the cell fusion experiments, came from Harvard, and now I believe they're actively trying to perform the nuclear transfer in the humans. We at the Whitehead Institute at MIT, where we work across the river, have also just sort of established a human embryonic stem cell lab. Our focus is not so much the technical aspects of the cloning, but we're just kind of getting started on trying to perform some of the genetic manipulations and just sort of better understand the basic biology of the human embryonic stem cell, because while mice and humans are 99.99% identical at the genetic level, the human versus mouse embryonic stem cells do present some differences in their handling and growth properties. And so one thing that we're studying in particular is the stability of the genome in the human ESLs over time. So people say that they can be passaged and self-renew indefinitely, but a lot of times if they're not really handled perfectly, you get all sorts of weird things over time. So chromosomes will break off from one another and fuse to one another, and extra chromosomes are gained when there's an improper cell division. So unless you're really an expert and you really know what you're doing, these things can sneak up on you. And after you've passaged a human ESL 10 or 20 times, you might be left with something that's totally useless unless you're doing very, very strict quality control. So what I think we're trying to, or what the human ESL lab across the river is trying to do, is really sort of establish the procedures for proper culture and proper normal maintenance of stem cells, and also introduction of genes and manipulation of genes, basically our knowledge in the mice that's been developed over the past 20 or 30 years, and translate that into the human cells.
And just to add to that, the Broad Institute, which is joint between Harvard and MIT, will be working on trying to differentiate these human ES cells into particular tissue types by adding different sorts of chemicals to these cells. So if you throw on a particular chemical, will that stem cell or stem cell line differentiate into beta islet cells secreting insulin, or will they become neurons? And so they're trying to do these differentiation assays using chemical assays, chemical screenings.
Let's try to go one last time to Worcester.
Cheryl, I think we're all set.
You're all set? Anything else in this room? Hold on just a second. I'll take one last question.
It's a good exercise running around that microphone.
What was the attitude in the research community towards the Korean research? Did people generally think, yeah, they did it, or was it like, well...
Yeah, people were convinced, and actually when the paper came, when the first allegations had come out, and the data was there for re-scrutinization, everybody was still quite convinced, and no one would really believe for an instant that someone could falsify something so
big and drastic.
Yeah, and because basically what had happened is after this was published, this fellow got money to set up human stem cell institutes, one in Asia, one in San Francisco, I believe, and the idea was that he was going to then pass on this knowledge to the research community, so that he would undertake such a thing and accept all that money, knowing full well that there's no knowledge to pass on, was really, I don't think anybody could ever imagine that. And the real trick is that the data saying that they have cloned the human, it's given the materials, it's fairly easy to validate. You just need the DNA from the donor and the DNA from the embryonic stem cells that you have, and some proof that they are in fact pluripotent embryonic stem cells, and then you can just do a simple DNA analysis to make sure they match up. But it's easy to test, but it's also very easy to falsify. So the scientific field is really one where we sort of take one another's word on their word. So it was a big shock, I think, to everybody. Any others?
Okay.
Any other questions?
I think we have a hand raised in Worcester. We'll take that as our last question.
All right.
Hold on for the mic.
Maybe it'll make more sense. Could you use a spectrometer to analyze a woman's embryonic fluid throughout her pregnancy to determine what growth factors were present at each stage of development, and thus were the genetic triggers to the differentiation at each stage?
So the growth factors that we're talking about may not be, I don't think you can detect with a spectrometer. The growth factors we're talking about would be genes that are turned on within the embryo itself, which such technology would not be able to detect. In order to see whether certain genes are turned on in that embryo, you would have to pretty much stain the embryo with antibodies against that gene, and for that you need to destroy the embryo. So currently we don't have the technology of identifying which genes are turned on in a live embryo in mammals.
So if I could add one thing to that, the coordinate, I can't see who I'm talking to, but the coordination of the development from the egg to something that resembles an organism is orchestrated by like thousands and thousands of proteins, some of which are transcription factors that are only within the nucleus of the cell, some are cytoplasmic proteins that are in the cell but not in the nucleus, and some are growth factors that float around outside the cell and bind to receptors on the cell surface. So I think it's a little more complicated than it might actually seem. But the good news is that there's been a lot of research done, and we do know a lot of growth factors that are critical. And for example, there's one or two that are critical for maintenance of stem cells that we know and that we add to our culture media to maintain the embryonic stem cells in their differentiated state. So, we maybe know 10%. I don't know. I'm just guessing at the number, but we know quite a bit, but we also don't know a lot. So.
Well, with that, I'll end the formal Q&A. And so at this point, please feel free to get up, get some snacks, and then talk to our researchers. I'd like to thank Worcester for joining us, and please join me in thanking our speakers tonight.